thank you for staying, and also congratulations to the organizers for making this very nice event. So I came from a Polymer Institute of the Academy of Sciences. So we are more involved in higher education. That means training PhD students, sometimes undergraduate students. But our spread of activities is very wide. So we do also summer schools and winter schools. This is more like for high school students. And we have then Polymer days or science days. This is for small kids at elementary school. So what I'm going to talk about is our new teaching model that we are developing. We got it also patented just in December. And this is it's called Elastic DNA Demonstrator, or EDNA. And basically, this is how it's called. We work in our research. We are focused on polymers and also polymeric materials, modeling. And we kind of discovered the space between existing DNA models and representation, which is not in existence, that those models do not show the relationship between deformations and structure of DNA. Okay, so why do I think the DNA teaching? So DNA is the most iconic molecule there is. So it's important for life. And we come into contact with it in on different levels. So not only kids are encountered with this, confronted with this in the textbooks, but we can see it in popular science media. We can see those on different icons and buttons, which symbolize science and research. There is another symbol which is very often used, is this planetary model of atom. This represents physical sciences. We know that this is not real. This does not exist. But DNA is real, but there are a lot of misconceptions about this. Now, so what are the most common misconceptions? So DNA is right-handed helix. And now, not only in visuals, but you can download 3D prints really from the internet. And they are labeled as educational models for biologists and for schools. And you print it, and it is wrong. It has wrong handedness. Then another another problem is the number of base pairs. So people do not understand that there is 10 base pairs per 1/3 of DNA, and 1/3 is this whole sinusoid, not only this one thing. So they try to stuff 10 base pairs here. Then there is also those models like difference between major and major proof, because those strands are offset a little bit. I will show it maybe later on. Now, there are, of course, there exist models which are perfectly correct. We can start with the most famous one, which comes from the work of Watson and Crick, who discovered the DNA structure. And just remind you, they didn't have plastics, so they couldn't build model from plastic. They didn't have computers. They couldn't make it in computers. So they had to really design it from scratch. They use metallic rods and spheres and plates, and they use kind of a scaffold, and which held these components. And now there are these physical analog models you can buy, which are structurally correct. Of course, they come to the price. A good model, one turn of DNA would be like 700 atoms. So an assembly kit would be priced, would be easily 200, 300 euros. But, and they kind of copy this concept that they are constructed around the scaffold. But what they miss is to show or to deliver the message what this structure implies. This is how the DNA molecule responds to external forces which occur in cell when there are activity of enzymes, and so there is exerted force applied on the DNA. So there are mechanical properties like bending stiffness, torsional, shear moduli, and tensional elasticity. Now, we do have these models we can make them in computer, but so we can download really exact structure, correct structure from crystallographic database, load it into computer, start molecular simulation. But this molecular simulation would take a lot of computer time, and the experience from it will not be so immediate because you have kind of apply imagination and transform it from 2D space into 3D picture of how this molecule behaves. So we wanted kind of to create something different. Okay, so what is EDNA? EDNA is elastic, what means reversibly deformable. So it's like a real molecule, it bends and springs back and so on. Then it's scalable. We can make it in different sizes, upscale, downscale, or make it longer, depending on which problem biology we want to focus on, explain. And reusable. Reusable in the sense of teaching that it can be used for biology, it can be used for chemistry, for physics, for biophysics, for polymers, and so on. So we want to build on these 3 pedagogical principles. One is the active learning. So the student would just play, touch the model like people do, and they immediately get a feel of spatial organization and a response of the molecule. Then this helps to build intuitive cognition. And then we have also this inquiry-based exploration where we provide this tool, this educational model with didactic aid, which collects some tasks for students and explanations. Okay, so just to recapitulate the main core structural features. So this is a structure which is downloaded from a crystallographic database. Then you see there is a minor groove, major groove. Then there is typical rise of the helix, and the helix is right-handed. Now, there are mechanical properties, and these mechanical properties we can measure experiments like atomic force microscopy, and we can recalculate them to design polymer materials like cross-linking and so on using chemistry. And we upscale this mechanical moduli to the sizes of our model. So these are, I will show 4 examples how we can use this model in the classroom. So since DNA is right-handed helix, so it has three-axial symmetry. So it will not, when you twist it in right-handed fashion, it will behave differently than when you twist it in left-handed. So when you twist it in right-handed, it will tighten. Left-handed, it loose. It will expose genomic information for activity of enzymes. And if it is tightened or twisted in different direction, this happens when you use, for example, antibiotics to address specialized enzymes, topoisomerases, and inhibit them. And the DNA tightens, and then the process is stopped, and the cell dies. So this is another problem in supercoiling. So we know that DNA is already coiled because it's double helix. And now when you introduce the twist, the torsional energy will be stored, and this thing will undergo, DNA will undergo spontaneously this transition when the DNA will coil around itself. So that's why it's called supercoiling. This is important for organization of genome in bacteria. Now, in higher organisms, in eukaryotes, there is additional mechanism of folding of DNA and condensating DNA. And so DNA is wrapped around proteins or histones and create this nucleosome. So we can demonstrate how DNA is organized in this nucleosome. And then there is problem of knotting. So DNA often, even from when it is ordered from bio companies for experiments. In 40% of cases, it comes noted. Those notes arise from activity of recombinases and during transcription and so on. And those notes can have chirality on themselves, which has an interplay between chirality of DNA. So this is what we can show. So we can show, we can go from biology to physics, how this bending and so on affects physical properties. And then we can go from physics to biology, like for example, when we bend the DNA because it is a helical structure, it will open mainly in the major groove, and there it will expose the genomic information. Now, in the first talk in the morning, there was also mentioned that there are new teaching tools which now involve AI and GPT. Now, so I would call for responsibility in this AI era. So I mentioned that there are a lot of misconceptions of DNA representation, and these appear also on a very renowned scientific magazine like Nature. Curiosity here is that the Nature was actually the magazine which published the original work by Watson and Crick with their structure of DNA. So this triggered an avalanche of letters to editor when they published this. Now, because AI is trained in this text to image generative agents to generate DNA, so I make a test with GPT, generate me visually appealing DNA, and in many ways the DNA is wrong. It's left-handed and so on. But it looks very nice, and I can imagine that a lot of students, a lot of people, a lot of popular media would use it as an illustration to their article. Now, this is another curiosity. So recently, I do participate in some TV, popular science cycles. So I was talking on TV about our research of polymers and DNA, and they added, I noticed it when I rewatched the episode at home, so they added into background those pictures, very nice, of DNA. All of them were wrong. So, and finally, when we were writing the chapter, so we noticed that also the DNA on the header is left-handed. But this one is good. This one is flipped. So this would maybe do first thing. Okay, so who can use EDNA? So this is, as I mentioned, so our outreach activities are very wide, and we built, EDNA is actually part of a larger project which we call LabBox, and it is an assembly of different experiments and demonstrators for students. And we have collaborations with universities, and we participate on this European Researchers' Nights and Open Door Days, seminars for students, summer schools, and so on. So what we think are educational benefits, so we think that playing with this model could improve spatial intuition, how DNA is organized, and to keep it longer and to remember it for a longer time. What could be future educational extensions? So we actually do have collaboration with university. We have requests. So they, for example, wanted to see this nucleosome. So we 3D print other structures and show how DNA wraps around them. Okay, so the take-home message would be that we face a challenge to stop the contamination of the virtual and media spaces, and we need to engage future generation of students to help us with this. So, and this is why it would be important to give them something to their hands. Okay, and this is just my contact information and some other research I should.
Thank you.